now let's move on to the next session. Let me introduce the chair of the next session. Is Dr. Xavier Saelens from University of Ghent. He is a professor there. He's a vaccine researcher. He is also a board member of ISV. So Xavier, please.
Welcome everyone to this session on recombinant protein vaccines. We have two speakers this afternoon or this morning or this evening for you. The first speaker is Dr. Chappell, who is an associate professor and group leader at the University of Queensland, and he has co-invented, together with Paul Young, the molecular clamp platform, which allows to stabilize recombinant protein subunit vaccines, that technology has been used for a number of viruses, including influenza, Ebola, and as we will learn in a moment, also for SARS-CoV-2. Dr. Chappell, the floor is yours.
Thank you very much. Thank you very much to the organisers, and I'm very much looking forward to presenting our data here in this forum. Next slide, please. So I thought to start off, I'd just give an overview of what we've been doing for this year. So we started working on this program the day that the sequence was released on January 12th. We partnered with Seqirus and received their adjuvant MF59 a month later on February 12th. We had chosen our lead vaccine candidate by February 14th, confirmed a strong neutralizing immune response by April 29th. We formed a formal partnership with a trusted manufacturer agreement through CSL. We achieved animal protection data in July, and then we commenced a phase 1 clinical trial on July 13th. So we've had a very busy year so far this year, as have all of the people on this call, I'm sure. Next slide, please. So our technology was funded by CEPI in CFP2 for epidemic preparedness. It's a platform technology for the manufacture of stabilized protein vaccines. The novel part of our system is the molecular clamp, which is a highly stable trimerization domain. It's a 6 helical bundle, and it works really well across a whole range of viral class 1 membrane fusion proteins. Some of the really special attributes of this platform is that we use a universal method of manufacturing in CHO cells and of purification using an antibody to the clamp domain itself. We've recently partnered with Cytiva to produce this resin at GMP quality in high yields. So it's working really well for scale up. Next slide, please. So at the start of the year, as soon as the sequence was released, the team began working extremely hard. We produced a whole range of modifications and changes within the spike protein of SARS-CoV-2, and we screened over 200 variants. Within 34 days, we had screened these variants for high expression and trimeric conformation. And the reason for doing this is we recognized really early on that manufacture would be crucial, and any advantages in yields we could produce at this early stage would translate to dose availability. Just to mention that the stabilization is facilitated by the clamp, and these antigens don't have the proline mutations that other platforms are using. Next slide, please. So once we had our lead candidate, we began optimizing the process of manufacture. We moved quickly from transients into stable pools and stable clones selected on the Berkeley Lights beacon. And by doing this, we were able to greatly improve the yields of expression, up to close to 500 milligrams per litre. So based on this production, we predict that we can achieve greater than 5 to 10 million doses per run in a 2,000 litre bioreactor. We've also shown some really promising liquid stability. So what we're showing here is the retention of the trimeric conformation. So any loss in this graph is due to aggregation or degradation of the product. And we see a really nice level of stability at 4 degrees and even at 40 degrees for 8 weeks. We still retain 50% of the trimeric soluble protein. Next slide, please. We've done some EM analysis of this protein and achieved a high resolution cryo-EM structure at 5 angstroms. We achieved this using the cryo-arm 300 that was recently installed in our facility here at the University of Queensland. Next slide, please. This protein has gone into mouse immunogenicity studies. We were able to show that with MF59 we induced a strong antigen-specific immune response. Apologies, this is a data-heavy slide. We've analysed the neutralizing immune response using 2 different assay methods. The first is the traditional microneutralization assay using TCID 50 measurements, and the second being an immunoplaque staining method. And for the immunoplaque staining method, we analysed NEUT response against both the original strain and the D614G mutant. In all cases, we were able to show a neutralizing immune response that was very strong with MF59. And what we're reporting here is relative to the NIBSC reference standard that's been supplied. So we can see in all cases, we generate between 2 and 6-fold higher neutralizing antibodies compared to that reference standard. We believe it's important to report the data relative to the reference, so all our benchmarking of the platforms relative to that of others, and we would encourage others to do so also. Next slide, please. So looking at the cellular immune response as well, we've done this using 2 assays. The first may be an assay that not everyone's familiar with. This is an in vivo based approach where we take splenocytes from a naive mice, label them with a fluorescent peptide array. We then take those splenocytes and inject them into the vaccinated mouse. And what we can do then is sort for the T helper cells, so we can directly visualize binding of the T helper cells to the antigen presenting cells. And here we show that MF59 elicits a strong T helper cell response. And we can also visualize direct killing of the splenocytes through the CTL response. And again, showing that our antigen with MF59 elicits a really strong CTL response. Next slide, please. The other assay, which is works really well and shows a very similar result, is using fluorescent staining and flow cytometry. Using this assay, it shows a very similar result, showing that MF59 with the antigen, it elicits a strong type 1 CD4 and CD8 response, including polyfunctional T cells labeled by interferon gamma, TNF alpha, and IL-2. Importantly here, we're showing that even in the BALB/C mice, we're stimulating a type 1 immune response. Next slide, please. We have also conducted a safety and toxicology study in rats. In this study, 3 doses of 20 micrograms antigen mixed with MF59 was administered. The findings, the doses were well tolerated with no adverse events. findings were limited to the muscle at the injection site and were mild and resolved during recovery. All of the rats produced a really strong antigen-specific response. And importantly, by analysing the IgG1 to IgG2A ratio, we could show that we had 50-fold more of the IgG2A, indicating a type 1 immune response. Next slide, please. Now, we've also undertaken a protection study using the hamster model. This was done by colleagues at Viroclinics in the Netherlands. The hamster model, we believe, is a really good model of this disease. In this model, we're administering 10 to the 2 TCID50 units, and you can see via throat swabs that the viral load peaks after 2 days and then decreases gradually. We have sacrificed animals at 2 time points, time point 4 and 4 days after infection or 8 days after infection. At 4 days post-infection, the lung titer is up around 10 to the 7 TCID50 units, and in the nasal tissue around 10 to the 7 to 10 to the 8. By day 8, this infection is mostly resolved. For comparison with the macaques, which have been used by most vaccine programs, for starters, 1,000 times more virus is administered to the macaques directly to the lung. And the studies that have used this model have shown that it's very difficult to recover any infectious virus after longer than 1 day after infection. So what you need to use is a qPCR-based approach. And if you compare the amount of virus administered and recovered, you're really only recovering less than 0.1% of the virus that's administered. So showing this comparison, I think it shows that the hamsters are a really superior model for analysing virus replication. Next slide, please. Also important for the hamster data is the pathology. So we can see a really good level of lung inflammation with significant parallels to the human disease. And importantly, if we look at gross pathology, on day 4, there's only around 10% of the lung tissue that's affected. But between day 4 and day 8, while the viral load is increasing, viral load is decreasing, we see an increase in the percentage of lung tissue that's affected. Next slide, please. So moving on to how our vaccine works in this model, we've conducted 2 studies. One was a single dose challenge study, and the next that I'll talk about is the 2-dose then challenge study. So here we can see that after a single dose of our vaccine with MF59, around half of the hamsters are completely protected in terms of virus in the lungs, and the other half have a decreased level of virus in the lungs. There's no significant decrease in the nasal turbinate, and in blue we're comparing against the inactivated virus with hydrogel. Next slide, please. If we look at the histopathology, this is where we really begin to see a differential between the S-clamp with MF59 compared to the inactivated virus. So looking at all the measures of severity in alveolitis and bronchitis, most significantly, we show a reduction in lung inflammation. Next slide, please. And here what we've included is some representative images. So you can see for the placebo and some of our hydrogel animals, there's a really strong inflammatory response. Some of the inactivated virus vaccinated animals do have a level of protection, but with MF59, a single dose, the mean score is 0.5, so a really good level of protection against disease in the lungs. Next slide, please. Moving on to the 2-dose challenge study. So we implemented a couple of changes when we moved to the study. We included a prior infection group that were infected with SARS-CoV-2 and allowed to recover. We also changed slightly the readout, so we culled half of the hamsters at day 4 post-infection and the other half at day 8 post-infection. You can see at day 4 we have a much stronger decrease in the lung titer, and in the nasal turbinate as well. We're starting to see a decrease in viral titer, but does not reach significance. And by day 8, the virus is cleared from both sites in the majority of animals. Next slide, please. Again, this is some data that I think is really interesting. So you can see moving from day 4 to day 8, the amount of the percentage of the lung that's affected by the that is negatively affected by the infection goes up, but with the S-clamp plus MF59, this is decreasing, and all of the animals show very low amount of lung tissue that is displaying disease. Unfortunately, we don't have the histology results from the 2-dose study to report tonight, but these should be coming in very, very soon. Next slide, please. So just to finish off, we've shown that the SARS-CoV-2 antigen S-clamp adopts the native conformation, and that we can produce it in a manner that's compatible with large-scale manufacturing. When formulated with MF59 adjuvant, the vaccine elicits a strong neutralizing immune response against SARS-CoV-2 as well as mutant strains. It also elicits a strong T helper cell response and cytotoxic T cell response, and provides protection both against viral replication and lung inflammation in the hamster model. We have a phase 1 clinical trial that's currently underway, and we are partnered with CSL, who are gearing up for large-scale manufacture to potentially supply this vaccine if trials continue to look as good as we're seeing the results as well so far. Next slide, please. So there are a huge amount of people all around the world that have contributed to this work. I'm showing here the group from the University of Queensland. Not too many people to mention, so just thank you to everyone who's contributed, and happy to take questions.
Thank you very much. Thank you very much, Keith, for this nice presentation. Am I live? I can't. So
you can go ahead, Xavier.
Okay, so thanks a lot for that very nice presentation and showing us this wonderful stabilization technology. What is the source of the clamp protein? Is it from a viral origin, and is there any chance that you would see a pre-existing immunity in the population?
Yes, so the clamp itself is derived from the HIV 6 helical bundle. Obviously there are people that are exposed to HIV, not a large portion of people. And we don't expect to see any negative side effects or negative implications from the inclusion of this domain in our vaccine.
Okay, thanks. There's another question raised through the chat. So why does the stability show a dip that recovers at the 2-week point? So somebody picked up that graph. The stability shows a dip.
Yeah, I guess you know, all graphs have a amount of error, so I wouldn't read too much into a slight dip. Yeah.
Okay. And there's another question from Ajit Singh. Would we need to vaccinate only immunologically naive individuals tested before vaccination? Not sure exactly what that indicates. So immunologically naive individuals.
So I guess the question is referring to, in general, do we vaccinate people that have been exposed to the virus already? You know, once we have a vaccine, I think that's a strategy that needs to be rolled out around the world. You know, vaccinating the most vulnerable people first and trying to contain the virus and eradicate the disease, and hopefully we can achieve that goal.
Okay, so time for one other question, perhaps. I noticed that the challenge dose in the hamster model was perhaps modest, 100 TCID50s, and in other studies, people have gone higher there. What was the reason to choose this particular dose of challenge?
Yeah, so what we wanted to do was we really wanted to rule out the potential for enhanced disease. So our strategy was to go for the lowest dose that could trigger some level of inflammation. And by doing that, if there was any enhanced disease, we would see that pushed further and more inflammation. What we saw was the opposite, that, you know, that hamsters aren't showing any inflammation in the lung, and we're seeing a good degree of protection after a single dose. So I think it's a really strong evidence that we aren't going to see enhanced disease with this vaccine, and hopefully with all the vaccines that are being produced.
Okay, well, thanks a lot once again, Dr. Chappell, for your contribution. We move on to the second speaker of this afternoon, which is Professor Dr. John Shiver, who is Senior Vice President of Global Vaccines R&D at Sanofi Pasteur. Before joining Pasteur, he was actually also heading the vaccine research at Merck, and throughout his career he has already contributed a lot substantially to develop novel vaccines, but also monoclonals against a range of diseases, including flu, RSV, cancer, Alzheimer's diseases. So he has a tremendous track record in those disease areas. Dr. Shiver, the floor is yours.
Thank you, Xavier, and thank you, Shan, and the organizers of the conference for this opportunity to speak today. Let's go to the next slide. Okay, so Sanofi is actually developing two different vaccine approaches for COVID-19, and one of them is a recombinant vaccine approach, a subunit approach based upon the S antigen protein, as everyone generally is doing, producing baculovirus, and with an adjuvant, which I'll describe a bit later. And we're doing this in collaboration with GSK for the adjuvant. And then the second is the mRNA vaccine approach, that is, that you've heard described, obviously quite a lot. Our candidate is being developed in collaboration with Translate Bio, utilizing the platform for mRNA and LNPs that they've been developing for over a decade, and in the context of our broader collaboration with them. Sanofi's rationale for two candidates was really given the importance and necessity of a strong response by the public health community and the pharmaceutical world against the COVID outbreak, to try to have a balanced approach in terms of a well-known, traditional, effective subunit vaccine approach, as well as a more novel approach, which we represent by the mRNA. And in our position as success by both of these, as well as all of the candidates that we're hearing, are going to be necessary in order to really contain this pandemic and provide the global supply of vaccine that's needed. Now, for the baculovirus approach, again, the platform we use has the advantage of being already licensed, both by Sanofi for our flu block vaccine, as well as baculovirus for other vaccine indications as well, notably HPV by GSK, for example. So we have existing large-scale capacity for manufacture, which is very important for being able to get the vaccine out. We have this for several reasons. One is because the flu block product that we have which is seasonal production and does give us the ability to utilize that capacity in order to produce this COVID vaccine. And of course, within the Sanofi overall production network, we have additional capacity that's going to be directed towards this vaccine as well. The BARDA is we're collaborating and being supported by, which is a huge boost to the program, of course. And we're collaborating, as I said, with GSK for their proven ASO3 adjuvant, the oil and water microemulsion, which has similarities to MF59 that was just described, and which significantly GSK has some very large stockpiles already from their pandemic flu efforts, and which actually really help enable implementation into a COVID vaccine. The timeline for this vaccine is phase 1, phase 2 start in September, actually projected for later next week, with earliest potential approval by next June. Referring to that capacity for production, the existing capacity we have, depending upon the doses selected, would be between 100 and 600 million doses. We're expanding, as I said, that capacity so that we should be able to produce more than 1 billion doses by next year. For the mRNA, as we know, it's an innovative approach. It's not a licensed product with it to date, but a significant promise with the technology that we are also very interested in exploring and learning its capability. And COVID gives an opportunity for doing that. The mRNA capacity for production and LNP selection has been greatly enhanced for a starting point by Translate Bio and their ability to produce these materials. We a little bit later, phase 1, phase 2 start, because there hasn't been a prior vaccine candidate by Translate Bio previously. They've worked for other purposes for therapy than vaccines. But because of the capacity investments they've made, they do project, again, depending upon the dose, 90 to 360 million doses available by 2021. And these productions, especially for the subunit, will happen concurrently with the development trials, as was outlined earlier by Linda's presentation. So next slide, please. So I'm going to start with the baculovirus expression system. Which again, briefly for those who don't know the expression system so well, the baculovirus is engineered to contain the gene of interest. You use that virus to infect insect cells, in a bioreactor in suspension, which is very advantageous. And then you purify the drug substance afterwards, formulate and fill. Again, no live pathogens. The baculovirus is completely removed and has extremely limited, host availability. You can pick the sequences. It's very flexible product design and is highly pure. It's considered to be a safe production system, and with limited adventitious agents, in the insect cells that it's produced from. As I said, cell suspension culture is available for this, and we can scale it to 21,000 liters currently, so given very, very large capacity and no biosafety containment requirements. From a regulatory point of view, the single cell line can produce multiple products, as evidence by our flu block vaccine, for example. And these products have been licensed worldwide using the platform. So it's really, really helpful that regulatory agencies around the world are familiar with this platform. So next slide, please. So, as everyone is doing, the spike protein is a major surface glycoprotein on the surface of the virus. It is the target for neutralizing antibodies predominantly. It is processed typically into S1 and S2 so that it can bind the human receptor ACE2. And of course, the S2 domain promotes fusion and cell entry. So next slide. The candidate that we're producing recombinantly would be the fixed, stabilized prefusion spike protein without the transmembrane region, so pre-S delta TM. And so to be produced as a secreted soluble protein. Again, squalene-based adjuvant in collaboration with ASO3, as I said, with GSK. AFO3 is a Sanofi Pasteur formulation that shares many similarities to ASO3, and we're testing both in parallel in our phase 1, phase 2 combined study that's coming up. The hallmarks of this type of formulation would be typically neutralizing antibody generation against the virus, as well as a more balanced Th1, Th2, CD4 T cell response. Next slide. So advantages of the platform, again, the manufacturing form is well understood by the FDA and other agencies. And if emergency use authorization is provided over the course of development of this and other vaccines, which should facilitate licensure. We can produce large quantities of vaccine, as I said, and AS03, fortunately from GSK and the work they've done, can be produced and provide at scale to fit the number of doses that I just said we project. Next slide. So briefly, the manufacturing process is that the virus is produced through basically up to four to five passages. It is added to the cells, which are been through several passages also to expand in fermentation and in suspension. It basically is purified by harvesting through centrifugation and extraction of the materials in the supernatant and clarified by a death filtration approach. Then several chromatography approaches are used to really purify the protein up to 90% plus, maintaining trimer, prefusion conformation, residual DNA is removed, and then finally formulated. Before formulation, one more filtration, and then combined with the adjuvant. Next slide. So these are the data that we've done in collaboration with the VRC, and the data shown here from Bob Cedars' laboratory, showing the humoral immunogenicity of this candidate, the pre-S delta TM with AS03 and rhesus macaques. And on the left, you can see the ELISA responses against the antigen, as a log 10 antibody binding titers, and as a course of 0 to 5 weeks. The immunizations are given as is becoming typical for programs with this virus at 0 and 21 days, and we have data out 5 weeks total. So about 2 weeks post the second vaccination. You can see from this that at the doses used, 5 and 15 micrograms of the protein with the AS03, the immune responses do start after the first immunization, but clearly increase upon the second administration without apparent dose difference between 5 and 15 micrograms in this species. Significantly, the AS03 is critical to make the immune responses. At 15 micrograms of the protein, as you can see on the far right, after 2 immunizations, there are really negligible ELISA responses. Functionally, using a pseudovirus neutralization assay, these are the 5-week data. These data are all quite new. So we'll have more time points and later time points later. You can see the 15 and 5 microgram doses in the adjuvant actually give very high neutralization titers in this assay, 3 log 10 titers, that really compare extremely favorably with a panel of human convalescent COVID-19 samples. In fact, they perform at the very top range of this large panel that the VRC has developed. And obviously the geometric mean well exceeding that by more than a log of that convalescent panel. And again, the non-adjuvanted protein did not show any neutralizing antibody responses. So for the subunit adjuvanted vaccine, these look like very promising data. And we'll continue these studies to follow them out, and a challenge is in progress and results should be available soon. Next slide. Confirmation of the expected T cell responses is that it does appear to be a mixed Th1, Th2 response. If you look at the memory CD4 T cells, are gated on CD4 R0 cells in total, you can see that the 2 formulations with the adjuvant do give, and this is a combination of IL-2, TNF-alpha, gamma interferon responses for both S1 and S2. And the responses are also seen against a representative Th2 markers, IL-4 and IL-13. Again, not to be surprised or unexpected for these type of squalene-based formulations. Again, these data came from Bob Cedars' lab, so in collaboration with NIH and GSK. So next slide. So the program timeline. The IND has already been submitted and approved. It takes a little longer to make a subunit vaccine because all proteins are distinct. Even though it's a platform, but there is some work that has to be done to optimize production of the protein and purification, but it's done. We expect to start the first subject in the phase 1/2 in September. It's a large combined phase 1/2 study, about 450 subjects, with the 5 and 15 microgram doses, and using both AF03 and our adjuvant Sanofi and the AS03 for comparison. And the phase 3, we should have the critical data in very early December, safety and immunogenicity data from the phase 1/2. So the phase 3 trial should start in December, and we expect that key phase 3 data could be available as soon as next May for licensure by early summer, potentially. Significantly, and industrialization has been starting in parallel and at risk of whatever the results may turn out to be from this clinical study. So drug substance production has begun really now, and then moving on to drug product formulation and production. And these are both phase 3 materials as well as eventual commercialization doses are being produced. Again, using the existing facilities that we already have, which I should mention, fortunately, do not impair our ability to deliver flu vaccine, which is critical also, because we all need flu vaccines this year. Just a nice little reminder there, especially with this COVID outbreak. So next slide. Now moving on to the mRNA approach that we're doing in collaboration with Translate Bio. Again, I think many people in this audience know about how mRNA is made in vitro by transcription, formulated with lipid nanoparticles, and can be injected directly as a vaccine, into humans. And the cell's own machinery turns the mRNA into protein, and that protein, becomes the immunogen. As I said earlier, Translate Bio has invested a lot in scale-up for producing this, largely because of their therapeutic protein demands, designed mostly for pulmonary delivery for therapy, and other indications, not vaccine, require large amounts. So they have existing ways to scale up to 500-gram doses per batch. So they're able to produce this at actually a fairly rapid timeline. It's cell-free, no infectious elements, and being RNA, no danger or risk of integration of the RNA into the genome of the recipient. Next slide. So briefly, this is a full-length S protein that's been encoded within the RNA, stabilized by the standard mutations that will fix this into a pre-fusion, non-cleavable form of the spike antigen. Next slide. So I think I've said most of this already. There is a self-adjuvanting aspect that is apparent with the lipid nanoparticle RNA formulation. So for immunogenicity, that may account in part for some of the strong immunogenicity that's been seen for this and other mRNA platforms. And this is highly scalable, as I said also. So next slide. Now this is new data that we have showing the immunogenicity in cynomolgus monkeys of our mRNA LNP of the spike protein. And on the left, you can see 3 doses of the RNA used at 15, 45, and 135 micrograms, the ELISA responses after immunizations, again at day 0 and 21. And you can see there's no apparent dose effect, and that after the 2 immunizations, there's a plateau of a fairly high, 10 to the 5th endpoint titers of binding antibodies at all 3 doses. And if you look at functional assays, this time in a microneutralization assay, that in fact you can see some beginning of neutralization starting after 1 immunization, but certainly substantially boosted after the second. A slight dose response perhaps at these limited time points. We only go out 42 days from day 0, which is only 3 weeks post dose 2. But again, comparing at all 3 doses very favorably with the convalescent serum set. I think it's 39 subjects that were collected for this, and all the monkeys either match the very highest titers of these convalescent sera or exceed. So we're very encouraged by the immunogenicity seen across these several formulations of the mRNA formulation. Next slide. If you look at the T cell phenotypes, as has been seen with mRNA and LNPs in general, it's very skewed towards Th1, as evidenced by gamma interferon production, and this is an ELISpot assay, and really not detectable Th2 with the represented cytokine IL-13 for the Th2 responses. Again, not surprising, and given the platform. Next slide. So partnerships are essential for success, and we've heard a lot about some of the partnerships already to date, and again. Government agencies, FDA, BARDA, NIH, Operation Warp Speed have been critical for supporting particularly our subunit approach, working with European agencies on supporting as well as access of vaccine supply to the different regions, scientific networks, and of course, a broad body of public health and not-for-profits that are participating as well. And a lot of collaboration between pharma companies, as evidenced by the GSK collaboration that we have, as well as the collaboration that we have with Translate Bio. Next slide. So briefly summary, we are working on two different approaches to help ensure success, for having a vaccine and more vaccines. Hopefully that will be successful, and also to determine what might be the best vaccine approaches. We're moving to clinical testing this fall. Both are manufacturable at scale, and we're going to, we're manufacturing at risk to make sure that supply of vaccine is available for broad distribution by the time we have data demonstrating safety and efficacy. Two immunizations preclinically do produce the highest functional antibody responses, as evidenced by neutralizing antibody titers, and the efficacy results preclinically are we should have soon, and we'll be showing those results as we obtain them. As I said before, the expected TH responses, by the type of formulation were obtained. And again, many critical partnerships, really, and a lot of collaboration, critical to advance these solutions at such a rapid pace. Final slide, please. And again, as was said in the previous slide, way too many collaborators and participants to name them all. But wanted to list the project teams at Sanofi Pasteur for both programs, the project team at Translate Bio, who've done such a great basis for building this platform, GSK's global project team on COVID, and the work they've done to make the AS03 adjuvant widely available, the people of the NIH, particularly the VRC and Bob Cedars lab, and the people that did the work to stabilize the pre-fusion form of the S protein, and for the funding support we received that's very significant from BARDA that really enables our subunit vaccine. Thank you all very much. So any questions at this point?
Thank you very much, John, for having shared these two parallel lines of development for COVID-19 vaccine at Sanofi and their partners. Perhaps I missed it, but do you have, about the protein vaccine produced in SF9 cells by Baculo system, are there any structural data or antigenicity data on the product as well?
Structural data, not yet. Cryo-electron microscopy is pending. And so we'll see what that looks like in terms of antigenicity with the antibodies we have right now. I think we can confirm the pre-fusion structure that we expect, and we do confirm that the subunit is largely trimer as expected as well. But further characterization is in progress.
Okay, good. So thank you. There's a question from the audience: can Sanofi formulate a combination of SARS-CoV-2 and influenza vaccine from the same platform?
I think it's certainly possible. It's something that we will explore. But right now it's important to show that the vaccine works per se, and we'll determine, along with the overall public health community, if it makes sense to do such a formulation. A lot will depend upon how endemic COVID tends to become. Many unknowns there, but certainly that potential exists.
Great, thanks. In the plan to license in May 2021 with immunogenicity data from phase 3, is that without efficacy confirmation by then?
That would be efficacy confirmation. The phase 3 that we're starting with the subunit vaccine in December will be 30,000 subjects. It would be 2 arms, placebo, and with the dose of the subunit vaccine being picked from the phase 1/2 study, which is a very considerable study, 450 subjects. So we should have a lot of data to inform the phase 3. We will collect efficacy data across a wide age range, and we will also collect it in people who have been established at baseline, whether they're COVID positive or negative. And we expect to be powered to show protection against acquisition of infection, as well as disease prevention.
Okay, perhaps the last question about the comparison between the protein approach and the mRNA vaccine approach. How do the two compare in terms of inducing neutralizing antibodies preclinically, I suppose?
In the monkeys, they look quite comparable. And of course, the key, as always, will be to see how these formulations perform in people. I'm very excited and interested in looking these data when they come about. As you could see in the monkey studies, because there were different collaborators, some of the assays were different. pseudo-newt, by the VRC versus a micro-newt that was done, elsewhere. In the clinical studies, we're going to be able to get a very good idea of how the two approaches do in fact compare head-to-head with the same types of assays by the same groups.
Mm-hmm. Okay, thank you very much, John. With this, I will close this session on recombinant protein vaccines, and we move on to the next session.
Thank you very much, Xavier, and also the speakers.